Welcome to this video on connective tissue proper. This video will introduce foundational concepts for understanding the structures and functions of connective tissues in the human body with a focus on the fibers of connective tissues. Cells of connective tissues will be covered in more detail in another video. Starting with an overview, it's important to know that there are specialized categories of connective tissue that include bone and cartilage, the solid CTs, and blood and lymph, the liquid CTs. However, today we are going to focus on connective tissue proper, which is found throughout the human body and serves to connect and support other tissues. Although not all connective tissue propers are composed of collagen, a significant majority of the body's connective tissue is composed of collagen. Indeed, collagen is the most abundant protein in the human body. So today we will use a collagenous tissue as the primary example of a CT proper. The collagen fibers found in connective tissues are produced by cells called fibroblasts. So let's have a quick peek at these cells before looking at examples of collagen itself. Fibroblast cells can be found in two different states, active and resting, and they appear quite different from each other. As shown in this schematic, active fibroblasts are cells that are busy laying down collagen fibers. Because they are active cells, they appear plump. Fibroblasts are only active enough to have this plump appearance during times such as initial embryonic development and during wound healing in adults. During most of our adult life, the fibroblasts are relatively inactive, and so they appear flat in most tissues of the human body. As shown here schematically, that means that only the nuclei are easily visualized, with the cytoplasms of the cells blending into the surrounding collagen fibers. Just before we look at an example of a light micrograph of this, you should take note that although the initial steps of collagen formation occur inside the fibroblast cells, assembly of collagen happens outside of the cells. Collagen is an extracellular protein. It is too large to be assembled inside cells. This explains why there is so much eosinophilia between the fibroblast nuclei in connective tissues. The eosinophilia is not the cytoplasm of the fibroblasts, but is rather large expanses of extracellular collagen. Let's take a quick look at this under the light microscope. Here's a high magnification section through a tendon which connects muscles to bone and therefore requires enormous tensile strength, which is accomplished by regularly arranged dense collagen fibers. Comparing it to the schematic that we just looked at, you can see the nuclei of the flat resting fibroblasts in amongst the large amounts of collagen. Now that you know about fibroblasts, let's look a little more closely at the collagen that they produce. As just mentioned, collagen, which is the most abundant protein in the human body, is produced by fibroblasts and is assembled extracellularly. But did you know that there are more than 16 different types of collagens? The types differ in their amino acid compositions and are located in different parts of the body. The most common types are types 1, 2, and 3. Type 1 is found in locations such as skin, tendons, and the organic components of bone. Type 2 is mostly found in cartilage, and type 3 is a specialized collagen known as reticular fibers, which are found in locations such as the bone marrow and lymph nodes. As I am about to show you, most collagens appear pink or eosinophilic under routine hematoxylin and eosin staining. Let's take a look. Shown here are three different light microscopic images that nicely demonstrate the three major arrangements of collagen fibers in connective tissues. On the left is what is classified as loose connective tissue because the collagen fibers are randomly arranged and not at all densely packed together. Because of this haphazard arrangement, loose connective tissue does not have much strength, but it is very useful for binding tissues together as well as allowing the diffusion of oxygen and nutrients across its structure. In the middle is what is classified as a dense irregular connective tissue. The fibers are quite densely packed, but they are not arranged in a regular pattern, hence the name irregular. This type of connective tissue is functionally significant in locations where stretching or deformity of the tissues is desired. Finally, on the far right is a tissue that you've already seen, dense regular connective tissue. The collagen fibers are very densely packed in together and are arranged in a very regular manner. This imparts tremendous tensile strength to the tissue and explains why this dense, regular connective tissue is located in tendons and ligaments. Now that you have been introduced to collagen, there are two other types of fibers that you will often hear about during microanatomical study of the human body: reticular fibers and elastic fibers. Let's first take a look at reticular fibers. Reticular fibers have a fun background story. When histologists first discovered them, they observed that they did not stain well with H and E, but were only nicely visible when stained with reticulin. So they thought that these fibers couldn't be composed of collagen, otherwise they would stain well with H and E, and so they gave them the new name of reticular fibers. However, with the advent of molecular biology, it was later determined that these fibers are indeed composed of collagen, just a different type, the so-called type 3 collagen. Reticular fibers are produced by reticular cells, which is just a fancy name for the fibroblasts that produce the reticular fibers. These fibers form a fine, spider-like web supporting mesh for cells in soft tissues, such as the bone marrow and lymphatics. This is shown schematically here, with the fine, spiderweb-like black fibers providing a supporting mesh for the cells that sit on the fibers. Now compare that schematic to a light micrograph, this one taken from a lymph node stained with reticulin. The black lines are the reticular fibers, and I hope that you can appreciate how they appear like a fine network of spider webs. The lymphatic cells sit on these fibers, but it is very difficult to see the cells in this light micrograph. The final type of fibers that we will consider are elastic fibers. These fibers are produced by fibroblasts and smooth muscle cells and are made up of linkages of elastin proteins. These are fun fibers because they are like physiological elastic bands that can stretch approximately 1.5 times their length. As schematically demonstrated here on the right, the fibers change from a very condensed squiggly pattern to a stretched, elongated pattern of arrangement. Because these fibers are the body's physiological elastic bands, it comes as no surprise that they are found in large blood vessels such as the aorta. The aorta is subjected to large pulsatile changes in volume as the heart beats. The wall of the aorta is able to accommodate these large changes in volume because of the elastic fibers in its wall. Shown here in this beautiful micrograph is a high-power view of part of the wall of a relaxed aorta. I hope that you can clearly appreciate the condensed, squiggly appearance of the elastic fibers, which appear pink in this section. These fibers will be able to stretch out as the volume of blood in the aorta increases with each heartbeat, and then contract again in between heartbeats. So the fibers act just like elastic bands, being stretched and then allowed to relax over and over again with each amazing heartbeat. This concludes this introductory video on connective tissue proper. Thank you for watching. You may access the virtual slides used in this video by clicking on the UBC Virtual Histo link in the banner of this YouTube channel.